Our next talk is Uros Kuzmanov from the Tony Gramellini lab, profiling of signaling cascade changes in heart pathology through phosphoproteomics.
Hello, everybody. I'd like to thank the organizers for organizing this event, and of course the Ted Rogers Center for giving me the funding to proceed with some of this research. So my background is, for the last couple of years, I've been in the lab of Dr. Andrew Emili. So it's the Systems Biology and Proteomics lab, which has had a fruitful relationship and collaboration with the Gramellini lab over the years, and this award has provided me opportunity to join Tony's lab as well, and I'm very appreciative. So today, even though the title is very general, today I'll talk to you about a specific example in our phosphoprofiling work, phosphoproteomic work on mouse model, the PLN R9C model of dilated cardiomyopathy. So, judging by the presentation yesterday, everybody, I think at least a couple of times this slide has been shown, and it shows the important role that Phospholamban plays in the electrochemical coupling in cardiomyocytes. And the PLN mutation is linked to a dominant disorder which leads to dilated cardiomyopathy. And this is recapitulated in the mouse model of the disease, which I'm going to be showing. And the mortality starts at 16 weeks of age for the mice. And there's been extensive work, proteomics work done by the Gramellini lab on this mouse model. So for us, the next logical step was to look at, as for proteomics people, to look at the phosphoproteomics of this mouse model. So we can tease out some of the signaling cascades which don't necessarily come out in proteomics experiments and profiling experiments. So why phosphoproteomics? 30% of all proteins are phosphorylated, and there's tens of thousands of phosphorylation sites on serine, threonine, and tyrosine residues. And as most of you know, these sites and these phosphorylation events are crucial in not only signaling cascades, but in other metabolic processes and enzymology and everything. So our normal proteomics pipeline starts with a mixture of proteins. 1/3 of those are phosphorylated. We digest these with the proteases, and then we get peptides, a mixture of peptides and phosphopeptides and other peptides that have post-translational modifications. So even though 30% of proteins are phosphorylated in our sort of proteomics pipeline, only 1% of the peptides are actually phosphorylated. Therefore, in addition to the mass spectrometry issue of identifying phosphopeptides that they ionize poorly due to suppression by non-phosphorylated peptides, there has to be a specific strategy and experimental flow how to get at phosphopeptides and identify these phosphorylation sites in any sort of context. So for this particular study, we had biological triplicates of mouse hearts from the R9C mice and wild-type mice. We went through the whole digestion process to increase coverage of identification. We also performed extensive hydrophilic interaction chromatography. And then from each of these fractions, we used titanium dioxide magnetic beads to enrich for phosphopeptides specifically. And then we did the tandem mass spectrometry identification of these, which can, due to their fragmentation patterns, we can identify the sequence of the peptide and the change in mass due to the phosphorylation. We can say where the phosphorylation site actually is. Once we had this information, we ranked based on quantified and ranked these hits, identified the phosphopeptides, mapped them to whole proteins, and then we did pathway enrichment to show which of these proteins play roles in different biological processes or signaling cascades, which are available in publicly annotated databases like KEGG, GO, Reactome. And based on these, then we will build an enrichment map and visualize it using a tool, Cytoscape, which I'll show later on. And from this kind of an experiment, we could tease out some signaling cascades which are dysregulated in any particular condition. Here, so this is what the raw data looks like. We identified a whole bunch of phosphoproteins. We also did normal proteomics workflow where we didn't do enrichment, and we identified 3,500 proteins here, around 2,000 cardiac phosphoproteins. And this is what our data sort of look like. On the x-axis, you can see the fold change of the proteins, and on the y-axis, you see the the p-value. And when we did set some cutoffs at 1.5-fold change for each individual phosphopeptide or protein, and the t-test value based on their intensities, in the identified experiments we found around 223 phosphopeptides with 251 phosphorylation sites that were significantly altered, and as well around 700 proteins. Then we tested these peptide sequences, these phosphorylation sites, to see what kinases, if there's a consensus sequence among these, a common sequence, and then which kinase is actually targeted. So, not surprisingly, some of the kinases which are central in cardiomyocyte physiology were identified like protein kinase A, GSK3, which was mentioned yesterday in a couple of talks, and the comogenin-dependent kinase 2, amongst others. So then what? So we have this list of proteins which were mapped from phosphorylation sites. So the issue with these publicly available databases is that there is no annotation for an individual post-translational site. So what we have to do to search the databases, we have to map a site and essentially ignore the site information, but use the protein identification to enrich for certain annotation terms, whether they be signaling or different biological processes within the cell. So we were dealing with these three data sets, the phosphoproteome, the proteome, and a merge set. So we had lists of proteins which we ranked based on p-value, and then from each of these we performed the gene enrichment analysis, which gave us then 409 sets, around 2,000 gene sets for the proteomic, 400 for the phosphoproteomic, and 2,500 from the merge set. And then, which I'll illustrate in the next slide, to further point out to further identify the gene sets or gene set enrichments or the biological process or the signaling cascades, whatever you want to call it, that were affected solely by the phosphorylation events, we only chose the proteins that were exclusively identified in the phosphoproteomic set or the merge set, ignoring the background set, which would assume that the phosphorylation changes that we saw in these proteins were simply due to the changes in the levels of the proteins rather than the alteration in the individual sites. So this is what these enrichment maps look like. So each node represents a gene set term from one of these databases. And as you can see, this is the global one, the one we would if we use all the data we had available from this study. And you can see it's kind of a mess, but we still see some of the crucial pathways and biological processes that occur in the heart. Like here, you can see protein kinase A activation, for example. But once we narrowed it down to these 804 pathways, which were phosphorylation sort of specific pathways, we emerged with a much neater picture. And from these, then we identified some just by literature searching, some pathways and general processes which have literature backup in cardiac sort of pathobiology. Out of these, one of the most significant hits was the notch signaling pathway. So just this is a very simple visualization of the notch signaling pathway. In general, a notch is a plasma membrane receptor. It has an extracellular domain and an intracellular domain. Upon binding of any one of its cognate ligands, it gets cleaved by ADAM proteases present in the extracellular space. Then it proceeds to the gamma secretase complex where it's cleaved, and the intracellular domain of notch can then go into the nucleus and activate transcription of target genes. So to validate, we performed a sort of a panel of Western blots, and we identified some changes in the processing enzymes which can overcleave notch and cause its inactivation. But most importantly, we saw that there's a very significant downregulation of the Notch1 receptor in R9C, and upregulation of processing enzymes, and some of the activating ligands are downregulated in R9C, and some of the inhibiting molecules which inhibit notch signaling in general were upregulated in R9C. So in general, given us, and the downstream targets were upregulated in notch signaling. So in general, given us a picture of notch suppression in the dilated cardiomyopathy, this particular mouse model. We then also further validated this with the immunofluorescence confocal microscopy. And here you can see notch in wild type and depressed in R9C. The ADAM extracellular protease upregulated R9C. And then as a sort of a control, we had notch 3 to show that this is a notch 1 specific effect. We didn't see any change. As well, then we further analyzing these. So these are cross sections of a ventricular tissue from these mice. We also did the nuclear localization quantification, and we also found that the intracellular domain of notch is, again, downregulated in the nuclei of R9C cardiomyocytes. And then this sort of gave us a sort of a simplified model, and this is so all of the proteins, so the notch pathway is much more complicated than these pictures suggest, but this is populated by the data we have generated. So this particular model is populated solely by the data we generated, whether it's from the phospho/proteomic experiments or from the classical microscopy and biochemistry experiments. And what we see, again, is a depression of the notch signaling in our model, in the mouse model of dilated cardiomyopathy. So as a general overview, we have identified in excess of 4,000 cardiac proteins, 2,000 of those being actual phosphoproteins. We identified around 4,000 phosphorylation sites, dozens of dysregulated signaling pathways, and we, of course, validated by independent means in sort of validating the whole approach, the suppression of notch1 signaling in dilated cardiomyopathy. So currently, and so in the future, we're looking forward to to a collaboration with Dr. Phyllis Bilia and Christine Rachel van der Laan, who is collecting some clinical heart tissue samples, which we want to perform this on, and we have started the analysis on some of these, and in the past we've also done some other mouse models and iPSLs and ES cells. So I'd like to thank, of course, the Ted Rogers Center for supporting this research, the University of Toronto, the Donnelly Center, where Dr. Amili's lab is, and of course the people from the lab, especially pointing out Hang Go, the mass spectrometrist in the Amili lab, and Ruth Esserling, the bioinformatician, and as well members of the Gramolini lab, which are present here, Diana, Jake, and Parveen is not here, but they've had direct contributions to this project. Thank you very much.
Thanks, Jos. We have time for a couple questions.
Hi. Slav Eppelman. Great talk, very interesting. I had two questions. So from the mass proteomics you take from the heart and dilated cardiomyopathy, do you have a sense of the proteins that are enriched? Which ones come from cardiomyocytes, which ones come from multiple cell types, and which don't?
So because we do the whole heart homogenates, there's no way. In our sort of global analysis, we don't pre-select for cell types, right? So we saw some beta cell signaling pathways. So obviously those are coming from some sort of an inflammatory response. So no, we don't, per se. For the notch, we were lucky enough, we had the cross sections, and we can validate it's actually in cardiomyocytes through the immunofluorescence. But in general, no. We could enrich for just taking ventricular tissue, which was suggested, but for us, the quantity of sample is also important. We need a lot of sample to actually pull out these phosphopeptides.
I had a quick.
The answer is no.
A second question. There's a paper published from my former mentor, Doug Mann, who has an inducible heart failure model where you can induce heart failure and then you release that, and there is remodeling, positive remodeling, reverse remodeling. And they showed reversal of gene expression patterns seen in heart failure. So these are genes. One of the criticisms of that paper was what's happening with the proteome. So in any of your systems, that's not really related to your work, but have you ever done a model where there's like a TAC model where you see heart failure and then you release it and you compare the proteome of heart failure and then improved heart failure?
There's been transcriptomic work on this done by, not by myself, it was done earlier in the lab. So in addition to all the proteomics work that has been done on this particular model, there's been also microarray work that was done a few years ago, and there was some, I don't know the details of it, but I know the general trend was apoptosis was playing a role, but I don't know, per se.
And one more question, Michelle?
Thanks. Michelle Bendak. Really nice studies. I'm just wondering, though, you're going to a lot of trouble to do the phosphoproteomics, and it really seems, unless I'm missing something, that you're just using it to tag proteins that you know phosphorylation is important in regulating signaling. Is there any intention to sort of look at relative importance of sites of phosphorylation or magnitude of phosphorylation and how that may contribute to disease process?
The issue is this makes these studies much more complicated. So the majority of the sites, the phosphorylation sites we identify, are not annotated anyway. So there's no antibodies for it. And most of the phosphorylated proteins that we're interested in are very low abundance. So even if we wanted to use a mass spectrometry method where we want to, measure particular phosphorylation sites, it would be very difficult in these complex samples. So because there's no way to enrich them. So and as I said, there's a lack of databases where we can actually say, okay, this phosphorylation site or this glycosylation site or this ubiquitination site, and then there's a whole bunch of annotations. This exists for protein level or gene level information, but for phosphorylation or any sort of post-translation, it doesn't exist. So we're forced back to map these things, and this is the best we can do in this sort of a global analysis. If we want to focus on something specific, which there's plenty of information there, it would take a lot of effort, generation of antibodies or generation of individual mass spec methods.
Part of the reason why I asked the question is just we've done some much cruder, much less elegant phosphoproteomics in my lab, and there's lots of sort of software, sort of database mining software that allows you to make some predictive, admittedly nothing experimental, but some predictive predictions about what phosphorylation sites might be important, what kinases might be mediating those phosphorylation sites, and I was just wondering if you have any intention to run your targets.
There was a table there that we showed on the significant sites which kinases are overrepresented for these consensus sequences which are enriched in our significant hits. So you can say that, okay, it's there, but you can follow it up. But I'm sort of a mass spectrometrist, so there's a lot of information there to actually follow up. But to choose it is difficult.
Okay, thank you.